This is lesson 4 of macromolecules, aiming at part B of the assignment, and we're going to be looking at the structure and function of carbohydrates in this session. It's very similar to the proteins section. In fact, it's identical in that we will be outlining the chemical structure of carbohydrates and then looking at what their role is in terms of membrane function. So, carbohydrate structure really is a little bit simpler, I find, than proteins. The basic fundamental building block of a carbohydrate is a monosaccharide. Obviously, mono meaning one. It's a single sugar, and you put two of those sugars together. Put two monosaccharides together, and you join them, and you get a disaccharide. Add one more to the equation, so you've got 1, 2, 3 monosaccharides joined together, you have a polysaccharide. And that's the general overview of a carbohydrate structure. Now these can be hundreds and thousands long, but it's still a polysaccharide. So let's get straight into the chemical structure of a monosaccharide. Now here we've got some that you may well have heard of already, i.e. fructose, galactose, ribose, and those that are switched on amongst you may well recognise that from RNA, ribonucleic acid. Even though RNA has a sugar phosphate backbone, and that's the sugar that forms the backbone of RNA, and the old classic glucose. So they're all monosaccharides, they're all building blocks of carbohydrates. Now, carbohydrates are named for a very specific reason. If you look at the chemical structure of these two monosaccharides, ribose and glucose, hopefully you can see from a casual glance at them that they're very, very similar in terms of their structure. We've got this carbon backbone down the middle. I should actually go one more up there. We've got carbon backbone all the way down the middle. Now, if we count the amount of carbons on ribose, 1, 2, 3, 4, and 5, which is what gives it the name pentose. It's a pentose sugar because it's got 5 carbons in it. And it doesn't take a genius to work out. Glucose is a hexose. Guess how many carbons it is? Well, I'll let you count them. It's obvious. The point that I was really leading to really was carbohydrates are called carbohydrates because each carbon atom is hydrated. And I'll say that again, each carbon atom is hydrated. So let's think about hydration. That means water, doesn't it? If you look at this first carbon here, not first carbon, but the first carbon that I've identified, you can see that attached to it, to the right and to the left here, you've got 2 hydrogens and 1 oxygen, H2O. So each carbon has 2 hydrogens, 1 oxygen. Each carbon has 2 hydrogens, 1 oxygen. 2 carbons, 2 hydrogens, 1 oxygen. And so you can see that it's where we get the name carbohydrates from, and it's the same here, here, and here, and so on. Now, what you can see down in this bottom right hand corner is that sometimes in textbooks and sometimes when you read in science information, you can see them described in a ring shape, in a sort of, I suppose, 2D shape, rather than in a linear shape like we've got up here with the chemical structure. We sometimes see it as a cyclical form, but don't worry about that. So hopefully from this sort of little slide you've got the names of monosaccharides, you've got some names down here too, and names across here, and you've got the basic structure in that they're a carbon backbone with H2O basically springing off it. and you've got a couple of groups at either end, a hydrogen at one end and a double bonded oxygen and a hydrogen at the other. So this is glucose, and you'll notice that it's changed from the first slide, and what we've got here is this is the linear form. This is the linear form. Yeah. And then this is the cyclical form. So you've got A and B. So this is linear, this is cyclical, which means it's which is a circle. Now, don't be afraid of this diagram. It's very much the same. However, all it means is that one of these corners here, 1, 2, 3, 4, 5, that's where the carbons are. And hopefully you can see H2O, 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 H2O. It's just the same molecule. It looks a bit scarier, but that's exactly the same molecule as what you saw on the left hand side over here. And this diagram I find is useful in the middle because it's showing you where it connects. So if you put glucose in a solvent like water, I believe, then this glucose mainly goes from a linear form into a circular form. So where it was like this on the left, if you put sugar in water, a lot of it goes to its circular form because this here bonds here. Hopefully that makes sense. So this will look hopefully quite familiar because it's exactly the same as how proteins join. What we're looking at here is how two monosaccharides join together. So on the top diagram here, we've got glucose and glucose, two monosaccharides, monosaccharide. And you should be very, very familiar with this kind of reaction. It is a condensation reaction or a dehydration reaction, sometimes called. And it means that water is being taken out. Again, it's the same reaction as we saw when we joined two amino acids together. But this time you can see where the water's coming from, hopefully, is this portion here. Why is water excluded? Because it is a happy atom. If you don't understand that, come and ask me. It's in there with my atom lesson and marshmallows. If you've not had the pleasure, I'm happy to go through it again. And when you take this water out, you can see hopefully that what is left is this oxygen atom here, which bonds the two monosaccharides together. Now, where we had amino acids, it was called a peptide bond. So when we joined two amino acids together, we formed a peptide bond. Well, hopefully you can see at this bottom bit here, when you join two monosaccharides together, you get a glycosidic bond. Pretty much the same thing. You're chaining two like building blocks together, and you're taking water out because you present them together, and water just likes to be made because the electrons. I'm not going to get into that, but that's how it works. So what we've got here is really the start of how simple sugars or simple carbohydrates can form to make a slightly more complex sugar. Here are a few more examples of disaccharides. So di meaning two, disaccharides. So here we've got glucose and another molecule of glucose, which forms maltose. Maybe you've heard of it. The second one down here, we've got monosaccharide, which is galactose. We've got a monosaccharide, which is glucose. Chain them together and you've got lactose. Quite an important sugar, particularly in dairy products, and you may well know somebody who's lactose intolerant. They don't have the enzyme that can break this down, so this causes problems later on in the digestive system, which again I'm happy to go into if you want to. Come and ask me. And finally, here in the bottom right, the third example, we've got glucose and fructose joined together to make a disaccharide and forming sucrose. So a couple of examples, throw one in for your assignment, and off you go. I should also mention, first of all, just recap that you can hopefully see this oxygen here, and this oxygen here. That is what's left of the glycosidic bond. That is the glycosidic bond, and hopefully you can see that in all three of these reactions, H2O has been taken out in a dehydration, synthesis, or condensation reaction. So when you start chaining 3 or more monosaccharides together, you get a polysaccharide, and each one of these sort of pentose, hexose sugars, should I say, is glucose. So glycogen is what's called a polymer. It means like a repeating unit, a repeating chain of glucose. So if you chain glucose together, glucose, then you get glycogen. Very important, particularly when we start learning about homeostasis and glucose homeostasis for the exam, in that if you eat a meal and you've eaten too much glucose and you need to store it, where do you store that? Well, you store it in the liver and the muscles as glycogen. So you actually, in effect, store it for later. So that has a consequence of reducing blood sugar. And when you miss a meal, when you skip a meal, or you're low on energy, what you can do is start chopping this all up, releasing it as glucose, and the cells can use it. Happy days, you've got energy. I threw this into the left here just because I wanted to show you that it's exactly the same reaction in between these sort of hexagons as what we've just seen. It's the oxygen in between the monosaccharides. We've got the glycosidic bond right there, and for each, let me circle it in red. Here's a monosaccharide. Here's a monosaccharide. In fact, is there 2 there? Yeah, there's. I really should have checked that. But it's definitely one there. I'm just questioning whether this is a glycosidic bond too. But anyway, I should have really checked that. But the key message is there, I'm not afraid to say it, is that each monosaccharide is joined by a glycosidic bond. And how do you do that? It's a condensation reaction. So we've hopefully followed it up to now. So now we're sort of talking about the function of carbohydrates on the cell membrane. This is a really nice little section because it should link a lot of knowledge that you got in cell biology. Now, I'd love you to read these 2 paragraphs in that, if we read this one first, proteins made by free ribosomes in the cytosol are almost devoid of carbohydrates. So the ribosomes that are floating around in the cell, in the cytoplasm, when they start creating proteins, what it's saying is there's no carbohydrates attached. Which isn't really surprising because these proteins will be used for the cell itself and they'll go on and do its job. However, nearly all proteins synthesized by ribosomes bound to the endoplasmic reticulum, so I don't know whether you can remember the rough endoplasmic reticulum or the RER, acquire carbohydrate units that are attached to them. So nearly all proteins that are made at the rough endoplasmic reticulum acquire carbohydrate units that are attached to them. And that's what we mean by modification of proteins. The proteins that come out of the rough endoplasmic reticulum are modified. They get added things, they get tweaked, they get like these superpowers, I suppose. And one of the ways in which they get these modifications is to add carbohydrates to them. So when we look at this fluid mosaic model, which you should be very familiar with from cell biology unit, you've got proteins embedded in the cell membrane. But now hopefully what you can see is these green chains coming out of the proteins and the lipids. So you've got a glycolipid and a glycoprotein. Glyco meaning sugar, and obviously the second bit would be protein and the second bit would lipid. So this is a sugar that's coming out of a protein, this is a sugar that's coming out of a lipid. And hopefully you can see now these glycomolecules, these hexagonal green discs that are on the diagram, basically are monosaccharides chained together by a dehydration reaction. And what you've got there is a complex sugar. This is a nice way to sort of see it in terms of a simplified diagram, I thought. You've got the cell membrane coming across with the protein embedded into the membrane and then the carbohydrates poking out of it, like so. That gives you more of a context about where carbohydrates are on the cell membrane, because that will lead us into what function the carbohydrates have in terms of the cell membrane. And again here I thought this resource was really nice in terms of saying 3 different things about carbohydrates on the cell membrane, because they have other jobs to do in the body, but the actual criteria of the assignment says what does carbohydrate, or what do carbohydrates do on the cell membrane, and I thought these things are perfect. So let's take this first one down here, which links to our work on infectious disease. Carbohydrates on cell surfaces may be important in intracellular recognition. Intercellular recognition. So basically, it's what we learn in cell biology. It's cells recognizing each other. And hopefully you can remember the sort of glycocalyx. The glycocalyx, the sugar coat, that's what it's describing. So my advice to you is go and find out about it. Dazzle me with your knowledge about what the carbohydrates do. Now I'm not talking about writing reams and reams and reams. I'm finding out quality citations that bolster that knowledge about how cells communicate with each other using carbohydrates. Secondly, carbohydrate groups may act as markers that determine the destination of a glycoprotein within the cell for export. So if this thing's being exported, it gives it a good marker as to where it might be going. Again, have a look into it. Can you tell me that you understand it? Can you tell me that you've read around it, and what citations have you got to say that? Keep them reliable. Books, journals, and with the odd website thrown in. And then thirdly, carbohydrate groups may help position or orientate glycoproteins in membranes. The carbohydrate groups prevent them from rotating in the membrane. So by by the fact that you've got carbohydrates poking out of them, if this protein decides to sort of roll away, it stops it from, misplacing or mispositioning on the membrane, and therefore keeps it doing its job. 3 things there that I think are understandable, logical, and sort of relatively straightforward, and things that we've learnt about already. So your task really for this assignment is to go away and make sure that you do a little bit of wider reading around it, and show me the quality of work of where you got your research from, and show that you understand not only the chemical structure of carbohydrates, which is well documented anyway. You can pick up any level 3 textbook and you can get lots of good sources from it, but it'd be really nice to get across your understanding of exactly what it does on the cell membrane. I hope this video has given you a good start and a good direction as to where to go. Any questions? Come and see me.